Hello students. Today's online lecture will be on the topics Physiology of Pregnancy and Parturition. This lecture session will help you understand about fertilization and implantation of ovum, pregnancy diagnostic tests, the maternal physiology in pregnancy and parturition process. The physiology of pregnancy is mainly concerned with the maternal adaptations that provide an ideal atmosphere for fertilization and nutrition to the growing fetus and a safe childbirth. The physiology of pregnancy can be discussed under the topics fertilization and implantation processes, maternal changes needed for adequate nutrition to fetus, and maternal changes needed for parturition and lactation. Let us discuss the topics one by one. Fertilization is the process of fusion of male and female gametes, that is, fusion of a spermatozoa and an ovum. The site for fertilization in humans is the ampulla of the fallopian tube. This process involves the events, namely transport of the gametes, sperm undergoing capacitation, and fusion of gametes. Before fertilization occurs, the ovum and sperms have to reach the site of fertilization, that is ampulla of the oviducts. In case of an ovum, after ovulation, it is expelled into the peritoneal cavity from where it is picked up by the fimbriae of the fallopian tube and sent to the ampulla where it waits for 2 to 3 days for fertilization to happen. During this time period, if fertilization occurs, then cell division starts immediately, whereas if there is no fertilization process, then the ovum dies. Now, in case of spermatozoa, after ejaculation, millions of sperms get deposited in the vagina. Within 30 to 60 minutes, they reach the fallopian tube due to their motility. The sperm motility is enhanced by the muscular contractions of the uterus due to release of oxytocin hormones and also due to other locally released hormones like estrogen and prostaglandins. Once the sperm reaches the female genital tract, they undergo through a process called as capacitation. Without this step, sperms are not capable to penetrate the ovum and fertilize it. It takes about 1 to 10 hours after ejaculation for the sperms to complete the capacitation process. During this process, the cholesterol part is removed from the spermatozoa and also the cell membrane becomes more permeable to calcium ions. Both these reactions makes the sperm more active. The next step is the fusion of gametes, which involves many steps. The first significant activity that occurs here is chemoattraction, where the sperms are chemically attracted to the ovum by the released chemicals. Then acrosomal reactions takes place that causes the sperms to penetrate the coverings of the ovum. The acrosome reaction involves the release of acrosin enzyme from the acrosome part of the sperm, which opens the penetrating pathway for passage of sperm head into the space between the zona pellucida and oocyte membrane. Only one sperm can enter the oocyte, and further entry of the sperms is prevented by activation of the ovum. Activation of ovum means when a sperm successfully penetrates the ovum, immediately the membrane potential of that ovum decreases, followed by some structural changes in the zona pellucida, and thus no other sperm can penetrate this fertilized ovum. The next major step during pregnancy is implantation of the embryo. It is the process of burrowing or attachment of the fertilized blastocyst deep into the endometrium of the uterus. The common site of implantation is the dorsal wall of the uterus. The first step during implantation process is the formation of a blastocyst. It is a 100-cell stage of the fertilized embryo. Blastocyst differentiates into an outer syncytiotrophoblast and an inner syncytiotrophoblast. This diagram shows the implantation process. You can see after fertilization, occurred at the ampulla, the fertilized cell divided and became a 100-celled structure known as the blastocyst. This blastocyst reaches the uterus within 4 to 5 days after fertilization and gets implanted in the endometrium of the uterus. Later, it differentiates into a syncytiotrophoblast and a cytotrophoblast. After the implantation, the endometrium is called as decidua. The cells of the endometrium gets enlarged, vacuolated and gets filled with glycogen and lipids to become decidual cells. These cells provide nutrition to the growing embryo till placenta takes up its functions. One thing to remember here is that in case of in vitro fertilization, the mature ova is fertilized with sperm outside the body and then implanted as blastocysts inside the uterus. The babies born by this method are called as test tube babies. Next is pregnancy tests. In an adult woman, amenorrhea, that is absence of menstruation, is the first sign of pregnancy, but it occurs in many other conditions as well. Therefore, detection of early pregnancy is made possible by certain pregnancy tests. These tests are based on the presence of human chorionic gonadotropin hormone in the urine of a pregnant female. The sensitivity of these tests varies and the concentration of hormone required for a positive test is about 2500 international units per liter. That occurs only at about 10 to 12 days after a missed period. The pregnancy detection tests can be divided into two types, namely biological tests and immunological tests. The biological tests are based upon the luteinizing action of hormone HCG and the immunological tests are based upon the antigenic properties of HCG hormone. Biological tests are namely Asham-Zondag test, Friedman test, Hogben test and Galli-Menini test. Nowadays these tests are not used. The second type of tests, that is immunological tests, are namely GRAVINDEX test, radioimmunoassay test and ELISA test. These tests are convenient and gives immediate results, thus are used more nowadays. The GRAVINDEX test of pregnancy is based on the principle that human chorionogonadotropin hormone antibodies can be induced in animals like rabbits and then the anti-serum thus produced can be used for the detection of HCG in urine or serum sample of a pregnant woman by using the techniques of complement fixation and precipitation. The GRAVINDEX test kit consists of GRAVINDEX antigen, GRAVINDEX antibodies and a dark colored slide. When a drop of urine sample from a pregnant woman which contains HCG is mixed with a drop of HCG antiserum, it causes neutralization of the HCG antiserum. If this is mixed with an HCG coated latex, no agglutination will occur because of the absence of antibodies. This indicates that the pregnancy test is positive and the female is pregnant. Again, in case of a non-pregnant female, her urine sample will not contain the hormone HCG. Therefore, if a drop of her urine sample is mixed with a drop of HCG antiserum, this will cause no neutralization. If this is mixed with an HCG coated latex, there will occur agglutination. This will indicate that the pregnancy test is negative and the female is non-pregnant. The next immunological test for pregnancy is radioimmunoassay. It is based on the principle of competition between a known radio-labeled antigen and an unknown unlabeled test antigen for a fixed amount of a specific antibody. Here, after antigen-antibody reaction, antigen separates into free and bound fractions and then their radioactivity is measured. The concentration of unlabeled test antigen is calculated from the bound and total antigen by using a reference curve. Another popular method for test of pregnancy is by using ELISA. It is enzyme-linked immunosorbent assay. It is a much simpler and sensitive technique and requires very small amount of the test reagents. It works on the principle similar to radioimmunoassay. The only difference is use of an enzyme here instead of a radioisotope. The enzyme acts on the substrate and gives a color reaction in case of a positive test. Pregnancy tests are important because they help in the diagnosis and confirmation of a pregnancy. These tests also assesses and determines the course of action in case of disturbed pregnancy. Pregnancy test is also helpful in early diagnosis of a hydratiform mole, which is a benign tumor of the placenta, and also for the detection of certain malignant tumors. Now let us see the physiological changes that occur in a woman during pregnancy. The normal average duration of pregnancy in humans is 280 days when it is calculated from the first day of the last menstrual cycle. As the pregnancy progresses, various types of extra demands occur from the mother's body for the growing fetus. These demands need to be met only by certain adjustment in the organ systems of the mother's body. The maternal physiological changes in pregnancy are the changes in the uterus, the changes in the body systems and organs, and metabolic changes. The uterus goes through various changes, and to accommodate the growing fetus, a marked increase in the size of uterus takes place. It increases in weight from 30 to 60 grams to about 800 grams or 1 kg. This enlargement is mainly due to hypertrophy and to some extent hyperplasia of the myometrium and smooth muscle fibers due to hormone estrogen in the early months of pregnancy. The total blood volume increases by about 30%. The plasma volume increases relatively more than the red cell volume, which leads to hemodilution. Therefore, pregnant female experiences physiological anemia of pregnancy. The total concentration of plasma proteins decreases from 7.5 to 6 gram per deciliter. The fibrinogen level increases and the serum albumin level decreases markedly. Total leukocyte count increases along with the increase in the clotting factors. The gravid uterus pushes the diaphragm upwards, resulting in change in the position of the heart slightly. The heart rate increases by 10 to 12 beats per minute. Also, the cardiac output increases at later weeks of pregnancy. Systolic blood pressure and diastolic blood pressure both decreases in the later weeks. Some females may develop pregnancy-induced hypertension. The vital capacity remains unchanged, whereas the body's oxygen consumption and the pulmonary ventilation rate both increases. This is known to be caused by the hormone progesterone. Some females may experience morning sickness like nausea and vomiting in the early phase of pregnancy. There is marked increase in the renal blood flow due to increase in the cardiac output and vasodilation. The glomerular filtration rate GFR also increases by 50% due to an increase in the renal blood flow and solute load. As a result of solute load, glycosuria is a common phenomena seen in pregnancy. Almost all the endocrine glands of mother react substantially during pregnancy. Thyroid gland shows mild enlargement due to increased thyroxine output. Pregnant female shows central nervous symptoms of cravings for unusual diets during pregnancy, and she also experiences alterations in mood and behavior. Hyperpigmentation occurs on the face called as chloasma and also on nipples, areolas and midline of abdomen during pregnancy. This occurs due to increased secretion of adrenocorticotropic hormone ACTH and melanocyte stimulating hormone MSH. The metabolic changes that occur are gain in body weight. A pregnant female can gain weight up to 12.5 kgs in average. Retention of water occurs in the second trimester of pregnancy due to decreased colloidal osmotic pressure and sodium retention caused by the action of sex steroids. When diet is balanced and adequate, then there is nitrogen retention in the body and positive nitrogen balance happens in a pregnant female. During pregnancy, the female stores about 50 grams of calcium and 35 to 40 grams of phosphorus. Total iron loss from the mother's body in entire pregnancy is about 500 milligrams, which should be supplemented by increasing the dietary intake of iron during pregnancy. Next topic is parturition. It is the process of giving birth to a young one, simply called as childbirth. It involves the preparation for the act of and recovery from the childbirth. From functional point of view, mechanics of parturition mainly involves uterine contractions and cervical dilatation. First, see how uterine contractions occur. The uterus, which remains quiescent during the period of pregnancy, becomes more and more excitable towards the end of the pregnancy until it begins strong rhythmical contractions that expels the fetus. Up to 30th week, the activity of uterus gradually increases. The female experiences Braxton Hicks contractions. These are periodic episodes of weak and slow rhythmic contractions which are painless and initially of mild intensity. Later, the intensity increases along with occurrence of pain just 1 month before labor. After 30th weeks, the uterine activity increases more. Throughout pregnancy, cervix remains rigid, but at the time of parturition, due to structural and biochemical changes, it becomes soft. This is called as cervical ripening. This allows the cervix to stretch when uterine contractions start and facilitate parturition. Parturition process occurs in various phases. The zero phase is under the influence of progesterone hormone. This phase lasts from the conception to the beginning of labor. The next phase, phase 1, lasts from the time of uterine activation to the onset of labor. Myometrium of the uterus becomes responsive to oxytocin in this phase and cervical ripening occurs. The phase 2 lasts from the time of delivery of the fetus to the delivery of placenta. And the phase 3 is the postpartum phase that persists till the restoration of fertility. In this phase, the uterus undergoes involution, that is, it goes back to its original state, and lactation occurs. About the details of the physiology of lactation, I will discuss in the next online lecture. Thank you.